We're going to learn about how we can react with them. We're going to understand how monosaccharides actually exist, not just as a straight chain.
This is a process that's unfolding. It doesn't start and say, here's one phase, and then stops and goes.
If I assume 100 grams, I'm also going to assume 100%, right? So the compound only consists of N and O, and it's
Hello, and welcome to another genetics theory session. What we're going to talk about today, and in fact in this whole section is about molecular genetics, we're really going to say is something that you might think already is obvious, that DNA is in fact the genetic material. Again, you probably know this by now, and a vast majority of non-students even know this. But most genetics courses are taught, there are some exceptions, in what we call a historical way. In other words, we start with Mendel. We talk a lot about chromosomes, meiosis and mitosis, gene mapping, sort of following along. And if you do it that way, as I say here, up until now, meaning you've done a lot of genetics with Mendel and chromosomes and things like that, but none of this has taken into account the chemical nature of genes. What are genes made of? And that is DNA. But those, you don't have to know that in order to do these Mendelian type problems, etc. And the people, Mendel and Sutton and all these other people we've discussed, did not know DNA was the genetic material. So it's very important, obviously, to find this out. Also, more broadly, people like Mendel had no idea how genotype determines phenotype, right? This is a very important concept, how genotype determines phenotype. He knew that there were two alleles, right, big P and little p, that made purple flowers or white flowers. But he didn't know what that really meant, in other words, that it was a DNA sequence. And he certainly didn't know how that allele made purple color actually come into existence. So these are the exciting questions, not that Mendel wasn't exciting, that we need to address in this entire section, all right? So the first thing we have to do is, again, we're going along historically and saying, how was it determined what the genetic material in fact is? What chemical in a cell is carrying this information that we know how to do stuff with, you know, in the Mendelian sense, and we know how to make predictions about what offspring are going to look like, but we don't really know what's going on at the cellular and more specifically the molecular level, okay? So before we can start thinking about this, we have to remind ourselves, what are the requirements for the genetic material? Just logically, what must be true about it? Well, certainly, it must be able to store and express information, right? There must be some chemical that has some variation in it, right? For example, we didn't discuss this, but you may know starch is just a repeating polymer of glucose, 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 etc. That's not a good candidate molecule for being able to store and express information, right? Information can be stored a lot of different ways. One you're familiar with today is in, say, a compact disc. It's stored optically in a digital format, a series of zeros and ones, right? So that's pretty simple. It's either 0 or 1, but there has to be some variation. If it was all zeros you had to work with, you couldn't encode any information. So that's one thing we have to keep in mind. We also know, obviously, that cells replicate, organisms replicate, so that means whatever the genetic material it is, it has to be able to copy or replicate itself, okay? And also, it must be able to allow variation. And I put mutation in there also, I'll explain what I mean. It was sort of implied by the first thing that we said, but since genetics seem to work the same way in every organism that anyone studied, from pea plants to fruit flies, etc., it seemed clear that there was so much variation in the biological world that the molecule in question must have massive amounts of potential variability, right? And we also knew, we being the scientists of the time, that mutation was one of the sources of this variation. We'll have whole lectures on mutation later. But just for now, realize we have to be able to account for the fact that mutations or changes can occur in this structure that would lead to variation. All right? So with all that in mind, let's look at what some people did to try and figure out what the genetic material was. When I say what do we know so far, I mean at the time, around the 1930s or something like that, what was in fact known on the biochemical or molecular front about genetics? Well, this isn't molecular or biochemical, but certainly one thing that we knew, Mendel said it, that a gene is somehow a unit of information, right? That we get. He said that it's a unit, one gene takes care of one trait, right? The P locus takes care of flower color, etc. So we know that. We also know that genes, whatever they are physically located on chromosomes. Again, that's not biochemical information. We know it from Sutton and from all the experiments that have been discussed recently. By the way, if your course is taught in a different order, you're going to have to go back and consult those things. When I say what we talked about before, you might say, we didn't talk about that before. Don't worry about it. You're going to get to that later and the connection will be made there. But most courses do go in this order. Okay, so here's the biochemical stuff. Biochemists analyzed chromosomes and they found out that basically there was about a 50-50 mixture in chromosomes of proteins and DNA. And it's very important to realize that around this time, before the 1940s, it was widely assumed that protein must be the genetic material. And I ask you the question, why here? Why would it be assumed that? Well, because of the great amount of variation, and because it was known that proteins were made up of amino acids, and there's 20 different amino acids, it seemed that proteins would have a potential for more variation. And actually, it's true, they do. DNA, there's only 4 bases. We're going to get to that in a minute. But geneticists and biochemists said that seems like there'd be less variability potential there. And proteins pretty much did everything. It was known that enzymes were proteins and other, functional things were proteins. So proteins were just sort of where it was at, and people assumed that proteins in some way would be the genetic material. Okay? Well, we now know they were wrong, but let's see the process. The experiment that's usually given credit for sort of starting it all is an experiment by Griffith, and this happened in 1928. Now, he didn't end with the conclusion that DNA was in fact the genetic material, but his experiment provided data, thought processes, and it was revisited later, as we'll see, as a way of an initial attempt to show that DNA was in fact the genetic material. So let's see what Griffiths did. This is an experiment that usually you have to know about in your course for a test or whatever. So Frederick Griffith, was working with bacterial pneumonia using mice, and this was in 1928, as I said. This famous experiment gave a clue to the nature of the gene, because he didn't really, when he wrote it up, he didn't conclude, yes, DNA is the genetic material. We'll see why. Well, what he knew is this. The pneumonia was caused by Streptococcus pneumoniae, which is still a common cause of pneumonia in mammals today. And he had identified 2 strains here, okay? He named them strain S and strain R. Now, what was the difference between the 2 strains? Well, the biggest difference was that strain S was virulent, in other words, able to cause disease, okay? So I say it caused death. Untreated pneumonia of the type that he was giving these mice usually did lead to their death. But the point was they got sick from this strain S. And if you grew it in a culture on a plate, the strain S cells appeared smooth and shiny, okay? Now, the R strain did not appear smooth and shiny. It appeared rough, which is where the R comes from. But more importantly, this strain was pretty much harmless if you inject it into a mouse. Did not cause any illness. Griffiths didn't know this, but we now know that the S